A tide pool looks simple, but it is one of the most intense habitats on the coast. It forms when seawater gets trapped in rocky basins as the tide goes out, creating shallow pockets that may be only inches deep or a few feet across. For a few hours, that trapped water becomes a separate world with its own chemistry, temperature, and biological interactions. Then the sea returns and reconnects everything. This repeated isolation and reconnection is the key idea. A tide pool is not stable space, it is a rhythmic environment that changes several times every day. That rhythm turns even small pools into ecological stress tests. Species living there are exposed to rapid shifts that many open ocean organisms would not tolerate. So when we study tide pools, we are not just looking at puddles on rock. We are looking at compact ecosystems where adaptation, competition, and survival play out in accelerated form. The driver behind this habitat is the tide cycle itself. Tides are shaped primarily by the moon's gravity, with the sun adding influence. In many coastlines, a full cycle is about 25 hours, usually producing 2 high tides and 2 low tides. As water rises and falls, the rocky intertidal zone repeatedly floods and drains. During low tide, water remains trapped in depressions, creating separate pools. During incoming tide, those pools are refreshed and reconnected to the larger marine system. This predictable but constant oscillation means organisms cannot rely on one set of conditions for long. A tide pool can behave like shallow sea in one period and like a stressed, evaporating basin in the next. The cycle does not pause for any species. Every resident must tolerate repeated transition between immersion, concentration, exposure, and replenishment. In ecological terms, tides act like a clock that continually resets environmental constraints and biological opportunities. Once isolated, a tide pool changes fast. Temperature can rise under direct sunlight, then drop when cloud cover, wind, or fresh tidal water arrives. Salinity may increase as water evaporates, then decrease after rainfall or new seawater inflow. Oxygen also fluctuates. Photosynthetic seaweed can elevate oxygen during daylight, while animals and microbes consume oxygen continuously, which may reduce available levels in warmer or stagnant conditions. Water depth declines as splashing and evaporation continue, concentrating organisms and dissolved substances into smaller volumes. These swings happen on short timescales, so survival depends less on peak performance and more on tolerance range. A species that thrives only in narrow conditions is at a disadvantage here. Tide pools reward flexibility, physiological buffering, behavioral timing, and efficient energy use. This is why intertidal biology is so valuable to researchers. It reveals how organisms function under repeated stress, not in controlled laboratory stability but in real environmental volatility. The organisms in tide pools are not random. They are specialists shaped by this instability. Barnacles permanently attach to rock and close their shell plates when exposed, reducing water loss. Mussels anchor in dense groups, which helps resist waves and lowers individual desiccation risk. Snails can retreat into shells and seal moisture inside during harsh intervals. Sea anemones contract into compact forms at low tide and reopen when submerged, balancing protection and feeding. Sea stars and urchins use crevices, grip surfaces, and exploit microzones where moisture persists. Small fish and crustaceans move through shadows, cracks, and deeper pockets as conditions change hour by hour. Seaweed is equally important, creating food, refuge, and localized chemical influence. Together these organisms form structured communities, not just scattered survivors. Their interactions are shaped by attachment space, water retention, food access, and exposure timing. In a tide pool, structure and behavior are inseparable from survival. Low tide also increases predation risk. As water recedes, many organisms become visible to seabirds and other shoreline hunters. Creatures that are protected by depth at high tide may be exposed in clear shallow water just hours later. This pressure favors defensive behavior, clamping down, blending with substrate, hiding in crevices, or reducing movement during vulnerable periods. Competition intensifies too. Attachment points on rock are limited, algal resources can be patchy, and shrinking water volume increases contact among species. Even wave energy, often viewed as damage, can be ecologically useful by redistributing nutrients, delivering larvae, and periodically resetting local dominance patterns. So the tide pool is not simply harsh because conditions fluctuate, it is harsh because physical stress and biological pressure overlap at the same time. Organisms must handle dehydration risk, changing chemistry, predation, and competition in repeated cycles. That overlapping pressure is what makes tide pools true natural selection arenas. Scientists have long used tide pools as high-value observation sites because they compress ecological processes into visible scale. In larger systems, many interactions are difficult to observe directly. In a tide pool, feeding, territoriality, shelter use, and stress responses can be watched in place with minimal equipment. This makes tide pools ideal for studying adaptation, zonation, species coexistence, and environmental thresholds. They also attract writers and philosophical thinkers because the contrast is striking. A tiny basin can reflect principles that govern much larger ecosystems. You can watch resilience, vulnerability, competition, and recovery in a space small enough to stand beside. That dual value, scientific and reflective, helps explain why tide pools remain culturally and academically important. They allow precise observation while encouraging broader ecological thinking. In practice, they function like miniature field laboratories where patterns of life are both immediate and profound, linking local detail to universal biological themes. Tide pools are also discussed in origin of life research as plausible settings for prebiotic chemistry. The idea is not certainty but suitability. Shallow coastal pools can provide repeated wetting and drying cycles, which may concentrate dissolved molecules that would be too dilute in open ocean. Mineral surfaces on rock can support catalytic interactions, and sunlight can provide energy that drives chemical transformations. Incoming tides can introduce new compounds, while temporary isolation allows local concentrations and reaction pathways to develop. Over long periods, those cycles can repeatedly test combinations of molecules under variable conditions. Whether life began in such environments remains unresolved, but tide pools fit several criteria researchers consider useful for early chemical complexity, concentration mechanisms, energy input, periodic replenishment, and reactive surfaces. Their value in this discussion is conceptual and experimental. They show how ordinary geophysical cycles can create chemical opportunities without requiring exotic conditions, making the emergence of complexity more plausible in natural settings. Another major hypothesis linked to intertidal systems concerns vertebrate evolution. Roughly 400 million years ago, the transition from fish-like ancestors toward land-capable forms required traits useful in unstable edge habitats. Tide pool environments offer a model for those pressures. Fluctuating water levels, temporary low oxygen, variable depth, and uneven rocky surfaces can favor organisms that tolerate short exposure, move effectively in shallow confined spaces, or use alternative breathing behaviors. Over evolutionary time, repeated selection in such variable zones could reinforce innovations in support structures, locomotion control, and respiratory flexibility. This does not mean tide pools were the only pathway, but they illustrate the type of environment where gradual terrestrial adaptation could be advantageous. Evolution often advances through repeated small benefits under recurring stress, not sudden dramatic leaps. Intertidal habitats represent that principle clearly. Each cycle rewards versatility. Studying these systems helps us visualize how environmental instability can drive long-term biological transition. Humans have sometimes modified coastal rock pools into safer seawater swimming areas by adding or reinforcing barriers that reduce wave force. In some locations, this is also associated with reducing risk from open water hazards, including large predators, by creating semi-separated bathing zones. These artificial or augmented pools are useful for recreation, but they are not ecologically identical to natural tide pools. Engineering changes water exchange, turbulence, and habitat structure, which can alter who lives there and how nutrients move. This comparison is important because it shows our dual relationship with the shore. We value the ocean's edge for access and safety, yet that access can reshape ecological dynamics. The lesson is not to reject all modifications, but to recognize trade-offs. When hydrology changes, biology changes. Natural tide pools are products of very specific physical rhythms, and even small structural interventions can influence species composition, stress patterns, and long-term habitat function. For education and public engagement, tidepooling is one of the most effective low-barrier activities in marine science. At low tide, people can directly observe organisms in their real habitat without scuba training, expensive gear, or boat logistics. That accessibility makes abstract ecology tangible. You can compare two nearby pools and immediately see how depth, shade, or wave exposure shifts community makeup. You can watch anemones contract, snails shelter, or crabs reposition as conditions change. This real-time feedback helps learners understand adaptation, niche use, and environmental stress more deeply than static images can. But good tidepooling requires restraint. Avoid stepping on organisms, turning rocks carelessly, or prolonged handling. The goal is observation with minimal impact, because these communities already operate near tolerance limits. Done responsibly, tidepooling creates both knowledge and stewardship. It teaches that coastal biodiversity is not distant wilderness, it is visible, local, and dependent on careful human behavior. In the end, tide pools matter far beyond their size. They are compact ecosystems where planetary forces, local chemistry, and biological strategy intersect in hours, not seasons. They reveal how life copes with repeated disturbance, how communities assemble under tight constraints, and how edge habitats can contribute to big evolutionary narratives. They also remind us that scientific insight often begins with close attention to ordinary places. A shallow pool on a rocky shore can illuminate principles relevant to resilience, adaptation, and environmental change at global scales. If we dismiss tide pools as leftover water, we miss their significance. If we treat them as living laboratories, they become one of the clearest windows into how nature builds complexity from recurring cycles of stress and recovery. Small in footprint, immense in meaning, tide pools show that some of Earth's most important lessons are written at the waterline. If you found this helpful, please like this video and subscribe for more explainers.